Hello students. Today in this class I am going to discuss about the rest of this of the portion of module 2, where I am going to discuss about DNA replication, RNA transcription and protein translation. That means it is the class where I am going to discuss about the genetic information flow. So first I am going to discuss the replication of DNA. I told you previously the DNA is the genetic material and it is lied inside the cell where the chromosome in the form of chromosome, and it reproduce. It is the only molecule, macromolecule, which has the power to reproduce itself, and this process is known as the replication. That means from one molecule of DNA, when it is replicated or when it is multiplied by 2, and then from 2 it will multiply to at 4, this process is known as replication, and it supply basically the information necessary to make all the RNA and protein in the body. And replication begins at the point in the DNA that is known as the origin of replication. And in this origin of replication, melting of hydrogen bonds are formed, and due to this type of melting of hydrogen bonds, y shape like structure will be formed, which is known as the replication form. So in the I told you that the genetic information flow, and this flow of this genetic material information in the molecular biology, it is known as the central dogma. Basically the information which is contained in the DNA molecule is expressed in the structure of proteins, and this is the total form of the central dogma, where here I am showing you that DNA has the ability to self replication, and when it transcribed into RNA, and from RNA, with the help of DNA, the RNA can be replicated, and also the RNA can flow its genetic information with the process of translation into proteins. The two strands of DNA in the double helix are complementary strands, and these are the antiparallel strands. When they are ready to replicate, the two strands unwind first, and then the bases in the cell wall migrate and bind with their complementary bases to form an exact replica of the original. That means these strand will be replicated first and form the copy of second strand. Also, this is true for the another strand, and the new complementary strands are formed on respect to the template strands. So the replication involves separation of two original strands, so what I have told you, and this separation can be done by the melting of hydrogen bonds, and it then synthesizes of two new daughter strands using the original strands as the template. The DNA double helix then first unwinds at the specific point, which is known as origin of replication, and then the polynucleotide chains are synthesized in both direction from origin of replication. That is, the DNA replication is bidirectional. First, the unwinding of DNA double helix. Replication of DNA starts with unwinding of the double helix. The unwinding can occur at either end or in the middle. The unwinding proteins are known as helices, and they attach themselves to one DNA strand and cause separation of the double helix. So here this is the pictorial diagram of DNA replication. Here I am showing you that DNA double helix replication is bidirectional. That means one strand is replicating in that direction and another one strand is replicating in this direction. Here keep one thing in mind, that one strand is replicating continuously, and whereas another strand replicating in a discrete manner. The strand which is replicating in continuous manner is known as the leading strand, whether this strand which is replicating in an discrete manner is known as lagging strand. And here the patch of newly formed DNA strands are known as the Okazaki fragments. Here the different enzymes those are responsible for DNA replication are shown in diagrammatically. Now comes to the next point, that is transcription. Transcription is the process by which information encoded in DNA molecule is copied into an mRNA molecule. It takes place in the nucleus. Also, DNA replication is a process that takes place in nucleus. The transcription starts when the DNA double helix begin to unwind near the gene to be transcribed. Only one strand of the DNA is transcribed, and this strand is known as the plus strand, and the another strand which is not transcribed is known as the minus strand. And the copy of RNA is just as it is the that is just it is like the minus strand. The ribonucleotide assemble along the unknown DNA strand in a complementary sequence. The enzyme which is required for the RNA transcription are called polymerases, and it basically catalyzes the transcription. Basically, the transcription is the information in one DNA strand that is transcribed into a strand of RNA. The termination site is the locus of the termination of transcription. So here we can here you can see that the mRNA is transcribed from the bubbling of DNA, or the replication or the unwound DNA double helix, from where the RNA polymerase are sitting, and it encodes the RNA on the template strand, and where the RNA is formed. The termination site is shown here. It is the sequence or site where the termination is where the transcription is terminated, basically. And the termination of transcription can be there is a protein known as raw protein which helps the process of termination in transcription. The first step of the transcription, the main main role of transcription is, it is the first step of protein synthesis, because protein synthesis can only be done on the strand of RNA, and from RNA only the proteins are synthesized. It is the first step where the DNA, where from the DNA the RNA are transcribed, and then on these RNA strands the proteins are synthesized. The segment of DNA that contains the necessary information unwinds to expose the bases. The exposed bases provide the template for messenger RNA, and it is known as the mRNA synthesis or transcription. Here, in case of RNA, the DNA bases which are present in the bases which are present in DNA are converted into the bases which are present in RNA, and in this manner the newly bases are inserted in the growing RNA chain, where we can found that adenine will base pair with uracil, and thymine will base pair with adenine, guanine will base pair with cytosine, and cytosine will base pair with guanine. And you can see that uracil is the new base which is only found in case of RNA and which is not present in DNA, as well as the RNA is devoid of any type of thymine. So, determining the sequence of bases in RNA, it is the basically the show that it is basically showing the sequence of bases in mRNA that would be synthesized from the template strand of DNA segment, which is the with the following sequence of the bases. If the template strand contains ATGCC AAGTG, then the non-coding strand is containing TAC, just the complementary of this this bases, and the base of RNA will be just as this one. Instead, the T you can write here the U. So the point is, the RNA strand is just the copy of the non-template strand, and it is devoid of any T, and instead of T you can write here the uracil as the base. So here the process of transcription is depicted in a short form. Now RNA, if we have the RNA, then it's the property of RNA to be transcribed into proteins. So the protein translation process is occurred inside the nucleus. after transcription, the RNA or the mRNA basically migrates from the nucleus to the cytosol, and where the cytosol, the ribosomes are present, and these ribosomes installed onto the mRNA, and from this mRNA, with the installed ribosome protein synthesis machinery, the proteins are synthesized. Basically, in this process of protein synthesis, all mRNA, rRNA, and tRNA, or these are all three types of RNA, are participating in translation process. Protein synthesis takes place on ribosome in cytosol. A ribosome dissociated into larger and smaller fragments, and these are different in case of prokaryotic and in eukaryotic system, and but the basic mechanisms are same. The small portion of ribosome which is known as 40s ribosome in case of eukaryotic system, are the portion where the mRNA can sit, and the portion of mRNA which is responsible for the ribosome recognition is known as the signed Algarno sequence in case of prokaryotic system, and it is also known as Kozak sequence in case of eukaryotic system. And after installation of the ribosomal small subunit onto the mRNA, then only the large subunit can sit, and it completes the total protein synthesis machinery. And after forming the total protein synthesis machinery, the triplicate of bases of mRNA, which is known as codons, are used for the insertion of charged tRNA. Charged tRNA means the tRNA which is containing the amino acid. For a particular amino acid, there is a particular tRNA, and when the charged tRNA comes, it can sit onto the codons, respective codons, and after sitting on respective codons, the translation is initiated. And this is the process where the total process is depicted in a short, and it contains basically 4 steps. And where the first step is the installation of ribosome, and second step is charged tRNA comes and sits, and after that it moves to next 3 bases or next 1 codons, and after that next tRNA with next tRNA which is carrying basically another amino acid for that codon comes and sits onto the next codon, and then only the chains are moving towards the 5 prime to 3 prime direction of mRNA, and by this process the translation process is going on. Basically, the mRNA lives in the nucleus and binds with the ribosome in cytoplasm first, and transfer RNA contains and anticodon contains an anticodon portion, which is a 3-base sequence that is complementary to the codons on the mRNA. Basically, tRNA carries the amino acids, and this is known as charged tRNA. The codon of the mRNA determines the amino acid sequence which will be present in the growing polypeptide chain. So the 4 steps of translations are activation, initiation, elongation, and termination. First, the activation. This means the tRNA should be activated or charged for the carrying of amino acid. This is the 3-dimensional structure of tRNA, and which has the anticodon loop, and this anticodon loop is a complementary to the codon of the mRNA, and it also has a CCA terminus which basically carries the amino acid. And when the charging of charged tRNA comes, it can stall into the ribosomal portion, and this is the small subunit, this is the large subunit, and this is the mRNA. And where the tRNA comes and sits onto the site of ribosome, where it binds with the codon anticodon complementary region, and due to this type of binding, the tRNA has the ability to transfer its charged amino acid onto the next growing polypeptide chain. Basically, the protein synthesis steps have 4 steps: the activation, initiation, elongation, and termination. In the activation step, the amino acid, ATP, tRNA, amino acid tRNA synthesis are involved. In the initiation step, the FMET tRNA, FMET, that means the tRNA which carrying the methionine, Translation begins only with the amino acid methionine, and that is why all proteins first amino acid is methionine. It is the main component of the initiation step, and it requires 30s ribosome, initiation factor proteins, and mRNA with the particular signed Dalgarno or Kozak sequence, which is responsible for the ribosome binding, and also it contains the largest subunit of ribosome and GTP. GTP will act as the energy source for the total process. Now, the elongation process includes the total ribosome machinery, the amino acid tRNAs, elongation factors, mRNA, and GTP. And the termination, when the termination step, it includes basically the releasing factors and GTP. The tRNA is specific for only one amino acid, and amino acid binds to an OH group of the appropriate tRNA by an ester bond. This OH group is present only on the CCA portion, which I have already shown you at this portion. And at the opposite end of the tRNA, this is known as the anticodon site or codon recognition site, and this triplicate of base aligns itself in a complementary fashion to the codon triplet on the mRNA. And this is the OH region site where the amino acid can bind with the covalent ester bond, and this is the amino acid, this is the anticodon portion, which is just opposite to the amino acid, and this is the basically activation of the tRNA. Basically, I am telling you that all the information are lied inside the macromolecules of DNA and RNA. Basically, these information are coded with the help of genetic code, and genetic code is the is a triplicate of bases which is basically determining the sequence of amino acids in a protein. And if the sequence is somehow disturbed, then all the amino acid sequence are disturbed, and thus genetic code is the primary step of genetic information flow. There are 64 codons, and 61 codes for 61 codons are basically code for amino acids. There are three remaining three are as the work as the termination codon, which are UAA, UAG, and UGA. The AUG is the initiation codon, which is coded by that methionine, the amino acid methionine. And there is a chance that more than one triplet can code for the same amino acid. Here is an example: leucine, serine, and arginine can be coded by more than one code. For 15 amino acids coded for the 2, 3, or 4 triplets, it is the only third letter of the codon that may varies. That means glycine, for example, is coded for by GGA, GGG, GGC, and GG. You can see that the third letter is are varying. This process is known as the Obel hypothesis. This third portion, third letter of the codon is known as the obeling portion, and changing of this portion may not be may not vary for the coding of any amino acid, and this is not responsible for coding of the amino acid. The code is almost universal, and it is the same in virus, prokaryote, and eukaryotes. The only exception is that some codons in mitochondria, which may have some another type of coding property, which may be vary, but rest of the codons are almost universal. So here, this is the process of translation elongation, where the mRNA on the mRNA, the protein synthesis machinery, the polypeptide is growing, growing one by one amino acid addition, and when the termination codons comes, that means UA, either UA, UGA, or UAG, then only the protein synthesis are terminated. And here some termination factors are responsible for this type of hydrolysis of growing polypeptide chain from the protein synthesis machinery. And when the all the growing polypeptide chain is hydrolyzed from the from the protein synthesis machinery, the ribosome also dissociate, also the tRNA dissociate, and from the mRNA. And after dissociating, the mRNA is degraded inside the cytoplasm. This is the process of steps of chain elongation process. This is the peptidyl tRNA, which is seated onto the which is sit onto the initiation codon, and after that the various factors are responsible for the elongation process, which are EFTU, EFTS, and EFTG, EFTU and EFTS, that requires GTP, and which is providing basically energy, and these are carrying the these are helping for the addition of another tRNA onto the growing polypeptide chain. And these amino charged tRNA, which are carrying basically particular amino acids, are joined with the peptidyl transfer process, and this peptidyl transfer process is basically the formation of covalent bond that is known as peptide bond. And these peptide bond formation basically are the key factor of the growing of the polypeptide chain. And after that, the translocation process is going on, which by which the ribosome moved for one codon, and it moves to the next codon, and this is the process by which the translation is going on. Now now come to the point, that is chain termination, and this chain termination requires the termination codon, either UA or UAG or UGA. These three are the termination codon which are found in the all type of organism. And there are some releasing factors that basically cleave the polypeptide chain from the last tRNA and release the tRNA from the ribosome, and this is also helpful for the releasing of polypeptide chain. So this is the release factor which binds at the ribosome, and it basically helps the tRNA to be removed from the protein synthesis machinery, and it also frees the polypeptide chain. And GTP now used for the energy, for which basically provides the energy to release of ribosomal subunits from mRNA. So translation is basically the information transfer, and this is the process by which the stored genetic material from DNA is transferred into the form of protein. This is the total summary of the class, where I have shown you that DNA has the ability to be replicated, and it has the ability to be transcribed into mRNA. And after the formation of mRNA, the translation is going on, which is basically the step of which is basically is involving of the protein synthesis machinery, and due to the translation process, the proteins are synthesized, which is basically the functional unit of life. So so students, in this class I have discussed about the genetic information transfer. Now come to the point, what are the mutation and mutagen? If I told you that if there is a any type of disturbance in the sequence, the whole polypeptide or whole protein will be disturbed, and this disturbance or error is known as the mutation. This is the error in copying of a sequence of a base. It can be occurred in occurred during DNA replication, during transcription, as well as during the translation. And due to this type of mutation, the different base or different amino acid can be inserted, and the whole sequence will be changed. And this is a non-heritable error. That means sorry, this if the change can be occurred in the copy during the copy DNA replication or transcription, they maybe have a chance to be to be recovered by, but if this mutation can occur during the transcription process, then it is very harmful for the protein structure. And if there is any type of structural dissymmetry found, or structural disturbance found, it is very harmful for the organism, because all the functions are related with the structure, and this is known as structure functional relationship. If the structure is disturbed, then also the function of the protein is disturbed. The mutagens are the chemical that causes the change in base, or the uh, 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 change in base or the amino acid. Basically, it is these are the chemicals, those are used for creating the mutations. There are many changes in base sequence caused by radiation, and these are all the mutagens. And these, if the base and nucleotides are repaired with this type of mutation, then this is called the nucleotide excision repair, or NER. NER is the process by which the mutation are prevented by cutting out the damaged area and resynthesizing the proper sequence. And basically, all not all mutations are harmful. Those mutations are not harmful, those are known as the silent mutation. So students, in this class I have discussed about the genetic information flow, and the mutations and mutagens in a short. So please go through these slides, and if you have any question, you can ask me later. Thank you.